Hello. Today we're going over mitosis and cytokinesis lecture. So we're going through the part of the cell cycle that is specifically mitosis. So you have the whole cell cycle, and a piece of it, like a piece of the pie, is going to be mitosis. At the start of mitosis, the DNA inside the nucleus begins to condense into tightly packed chromosomes. So we're going from kind of like a spaghetti arrangement, it's often called, of DNA, where it's loose, to tightening it up, condensing it down, and pulling it together. Before this stage, the DNA exists as loose chromatin that allows the cell to read genes and make proteins. So this is the time that it's normally using the DNA in the cell, is when it's not going through mitosis and replication. The chromatin wraps around proteins called histones, which help organize the DNA, which you can see here. There's these large complexes that the DNA wraps around to keep it organized. As mitosis begins, the chromatin coils more tightly, so each chromosome becomes visible under a microscope. So you can actually, this is a chromosome here, a replicated chromatid, and you can see these under a microscope because there's so much DNA that's all packed and condensed down into this structure. After DNA replication, each chromosome is made of two identical chromatids. So the DNA before mitosis was already replicated. Here is one chromosome. This is its duplicate, the exact copy, okay? The two chromatids are attached at a region called the centromere here. The centromere will later serve as the attachment point for spindle fibers that separate the chromatids. So we've got a copy on one side, a copy on the other. Eventually, we're going to separate and pull those apart. This organization ensures that each new cell receives a complete set of chromosomes. So every chromosome is going to duplicate itself, and then we're taking the duplicated pieces and putting them into a separate cell, where the original ones also then go into another cell. All right, let's continue. The ends of every chromosome are protected by structures called telomeres, which you can see here up at the top, the telomeres. It's just an end cap that signals the cell a few things, mainly that the end of that DNA strand is there. It kind of stops things, machinery and DNA replication at those points, so it doesn't just try and keep going or anything like that. Telomeres help prevent the chromosome ends from being damaged or sticking to other chromosomes. During chromosome condensation, the telomeres remain at the tips of each chromatid. These protective caps are important for maintaining the stability of genetic information. Okay, so we're just wrapping this up. This, remember, is the point where we've replicated the DNA, and now we're condensing it down into a form that can be used in the next stages of mitosis. Condensing chromosomes makes it easier for the cell to move DNA without tangling or breaking it. The tightly packed structure allows the spindle fibers to pull each chromatid to opposite sides of the cell during mitosis. Once cell division is complete, the chromosomes uncoil back into chromatids so the DNA can be used again. So it uncoils from this very highly structured, and remember, at that point you're only talking about one. It uncoils from its highly organized structure into a much looser structure so that the genes on the DNA can be read and utilized. This careful process helps produce two genetically identical daughter cells. So again, the point of all this was making a replica cell, going from one cell to two cells. Okay, now let's look at the individual stages of mitosis. So here's our whole cell cycle, where you get cell growth happening as it prepares to replicate itself. The DNA gets replicated. We prepare for mitosis with that, with the DNA becoming condensed, right? Now, this yellow section here has the different phases of mitosis. This is what we're going to talk about. And then this last stage, which is cytokinesis. Okay, mitosis is the process by which a cell divides its duplicated chromosomes into two genetically identical nuclei. The process has 5 main stages: prophase, metaphase, anaphase, telophase, and cytokinesis. Each stage involves changes that help ensure the chromosomes are separated correctly. So there's specific things that happen at each one of these stages that can tell us what stage we're in, what part of the process we're in. Together, these stages allow one cell to produce two daughter cells with the same genetic information. During prophase, so we're starting with the first phase right here. During prophase, the chromosomes condense and become visible under a microscope. We saw these, right? Like if you look at this red X, remember, this is one chromosome here, and this is the copy of that chromosome here. Well, now we can see them under a microscope once they're all condensed and they look like these X's. The nuclear envelope breaks down, which you see in purple, this circle that has, that's dashed. It's got spaces because that envelope is breaking down. And spindle fibers begin forming from structures called centrosomes. During metaphase, this is the next one now, you can see the differences between these two pictures. The chromosomes move towards the center of the cell and move towards the center of the cell. So you see that here, they're lined up in the center. Spindle fibers attach to the chromosomes and line them up along the middle of the cell. So that's what these lines are here. These are spindle fibers, and they're attaching to the chromosomes and putting them in that middle line. They're putting them in that orientation. Okay. During anaphase, the sister chromatids separate and are pulled towards opposite ends of the cell. So remember, they were lined up on this middle line. The spindle fibers start to pull them back. And as they do that, they separate and pull away from each other. Each separate chromatid is now considered an individual chromosome. So this green one here is an individual chromosome. There's a duplicate of that green one over here. That's the same chromosome duplicated, like a copy. During telophase, the chromosomes reach opposite sides of the cell and begin to uncoil. So we see that here. New nuclear envelopes form around each set of chromosomes, creating two separate nuclei. So now we're forming an envelope around these chromosomes, new envelopes around each of these two. And you see we were at one cell, and now we're separating into two cells. The final stage, cytokinesis, which is here, divides the cytoplasm and separates the cell into two daughter cells. In animal cells, the cell membrane pinches inward to form two cells. So pinches inward, it just keeps going in this direction until finally it's barely together, and then it pinches off and squeezes into two separate cells. Each daughter cell receives an identical set of chromosomes and can begin carrying out its normal functions. So at this point, it's just a regular cell. Normal cell doing whatever that cell is supposed to be doing. The 5 stages work together to ensure that genetic information is accurately distributed during cell division. You probably have to review this a few times to get comfortable with it because there's a lot going on, but it is definitely something that makes logical sense. You can follow the steps one by one to see what they're doing and what the purpose is. You just have to spend enough time with it to remember what that is. Let me know if you have any questions. Thank you very much.